Hello?
Hi.
Hi,
Hello, sir.
Hello? Hello? Lalith, Soham, Jayadratha. I didn't know if someone said that they might want to present on what they were working on. If that's true, you're welcome to present today.
Yeah. Can I present today?
Yeah.
No. Should I wait or should I share my screen and start? Should I wait for some time?
No, we can go ahead.
Okay.
Looks good.
Okay. Yeah. I'm Jayadratha. Currently, I'm an MA student at IIIT Hyderabad. Today, I'll talk about an introduction to temporal graph and their neural networks, the unveiling time in networks. So, yeah, so the outline of the talk will be, at first I'll talk about graphs and networks as a whole and I'll introduce it. And then I'll talk about temporal graphs, how it captures the time. And then I'll talk about uh, how neural network can be designed for temporal graphs. And there are two different kinds of temporal graph. One is DTDG and another is CTDG. That is discrete time temporal graph and continuous time temporal graph. And I'll talk about, for each one of them, I'll talk about one model. Those are very popular for CTDG and for DTDG, it is evolution. Then I'll talk about applications, how we can use it for DevoWorm and C elegant development. I talk about my thought only here and I conclude. So why networks? We use networks to understand relations. So graphs, those are very powerful to represent relationship between entities. If there is some citation graph or social media graph, or it can be some road network, we can use to understand the relationship. So think of some social network or biological interaction. Here, the entities are nodes. That it has entity and relationship. So we use nodes as the entities and age as the relationship. For social media, we use people as the node and friendship as age. For citation network, we use papers as the citation, paper as the nodes, and citations as the age, like that. So at first I'll talk about the simplest of all, that is static graph, and I'll talk about it's how we use graph neural network to to work on those static structure. So let's take a molecule here. So suppose we are taking nitrogen, and here what we do is, for this graph, we can represent it as a graph, and for nitrogen we have different what you call it, different neighbors, and those neighbors are different atoms, and we can use here for each uh, for each layer we have their neighbors, and this is neighbor of neighbors, and these are the neighbor of neighbors. And in each layer we use some kind of neural network here. Here we use some neural network, and those are shared parameter neural network. This is the GNN, overall GNN structure, and here we do message passing. So to update this nitrogen, we pass the features of of its neighborhood as messages, and we will update in that way the overall features. And GNN can learn patterns and make prediction based on the graph structure as well as node feature. So instead of MLP or any other basic neural network, we use GNN just to capture its graph structure. So there are three different kind of tasks that can be designed here. One is node level task, edge level task, and graph level task overall. And for all of those, sorry. And you were telling something?
No, I think you're fine. Go ahead.
Okay. So for all these kind of tasks, we have we can have classification as well as regression. But in reality, we have like dynamic. That is, it is changing with time. Not always static graph can represent all kind of networks. Suppose there is a road network. So if you see the road network, it is constantly changing. So how can we capture that when network is evolving with time? So here relationship might appear, disappear, or strength might change. So here what we can do, we can capture only the snapshot of the time for us some time. But if we just take a snapshot and use GNNs or some other kind of neural network, the problem of that will be we can miss some intermediate all those temporal information, crucial information. So the example can be for social interaction or financial market fluctuation or biological system development. Here it is changing all the time. It can be daily, hourly, or in every moment. So now we understand why we need temporal graphs. So now we'll take detailed look into it. So what it can capture. So they capture how relationship change over time. So here for this graph, at different time there can be new edge addition or new node that might come. Some new node has come, some new edge might come, or node update might happen, or edge update might happen. So here the key idea will be for different time step or time interval, at different time interval, a node and edges might change. So it's a dynamic system. So here we have to understand the temporal pattern as well. So only capturing its spatial features will not work here. So we need both temporal as well as spatial feature. If we see in terms of model complexity, for static graph, the models are easier. So if we have age-weighted, where the age might change, suppose for a network, for a social media network, the friend I have with whom I am talking every day, his feature or her feature might be more relatable to me. But someone I never talk to, his or her feature will not be that important. So here the weight will come, and model complexity will increase with discrete time, and for continuous time it will be the maximum. When continuously with time the graphs is changing. So for how the data structure look for temporal graph. So for temporal graph, as we see at time t1, there is a edge between two nodes. and at T2 there might be some more age that is coming, or more nodes coming. And at T3 there are more nodes in temporal age that can be represented as at source node, then target node, when there is a time like age that is appearing and its timestamp. This is how we see temporal graph. Instead of static graph, we use temporal ages or events. Instead of only the node we take, we can have multiple interaction between the same node also, that we have to capture. And each event represent an interaction that or relationship that occurring on a specific time or some time interval. So there are two kind of temporal graphs. One is discrete type temporal graphs, that is the snapshot based. Suppose this is a graph, we take snapshot at different time, and as we see the structure is changing. We just take at different time step at a time interval. Suppose we are taking a time interval daily, hourly, or some time interval t. We can see how it is changing at different time. We just take the snapshot. So here node or age can appear or disappear, or it might be same as well. So we are taking a discrete time. That's why it is discrete time temporal graph. So example I have shown here, one example of DTDG, that is social media, social network evaluation. So it's a very small and simple graph I have shown. So for 3 months, suppose every month I am taking a snapshot. So these are the friends A, B, C, D, and E. So A is friend with B, B is friend with C, C is friend with D, and there is no friend of E at the month 1. At month 2, I am taking a snapshot. So what happened here? So A and B, the friendship is there as it is, and between C and D also there is a friendship that is there as it is. There are new connection that has been made between A and C and B and D, and the friendship that this broke between B and C, and here also it has changed. So each month we have taken a snapshot of the static graph. So together they form a discrete time temporal graph. So now I'll talk about continuous time temporal graph, where we see event that as it is happening. So what is happening? Suppose I am taking a Twitter graph. So this person is tweeting at this timestamp. So we'll take a snapshot. Then after some time, there is another person who is tweeting at this timestamp. So then again another person who tweeted like that. That as time goes, there is some new ages that is coming up. So based on this, can we understand what will be the future age, if there will be any future age? So here there are both kind of interaction between users and the content. So this person can retweet this person's tweet. It might also happen. So here the catch is no fixed time interval. Whenever there is some event is happening, we are taking a timestamp precisely at that time, and so here we are getting more fine-grained representation or temporal dynamics. Where it is useful, so for financial transaction or online interaction, we can use these things. So example is CTDZ in financial transaction, where in seconds or in milliseconds, there are millions of transactions that is happening. So CTDZ can capture those exact timing of each transaction. Where it will be useful? So it will be useful for fraud detection or analyzing market dynamics or recommendation system we can use. So at time t1, there is this graph. At time t2, there is this, both of these is happening. One new age that has come, and one new node that appear. And at T1 there is new node appear, in T2 new edge appear, and T3 there is another edge appear. And this was at first at T0. This was the graphs for a transaction. So new node, that means for a financial transaction there is new user that has come. And suppose this is a user and this is a merchant. So if this person has buy something at T2, so there will be a connection, and similarly there will be another connection here. So this is CTD. So between CTDG and DTDG, which one to choose? So as I have told, CTDG represent more fine-grained timing, and discrete time temporal graph or dynamic graph, it represents the snapshot at different time. So every 30 minutes, here I have shown for a Twitter network. Every 30 minutes, I am taking a snapshot. So we can see here, DTDG is more, it is simpler, easier to process, and good for data, naturally aggregated into timestamp. But the problem with it will be loss of information in intermediate steps. If there are new edges or something is coming, or again it go away, then we just we cannot track that. We just miss that information. For CTDG, it will be more accurate. It captures fine-grained temporal information, but problem is it is more complex to process. So here it will be essential when there will be precise event timing we need. So its choice depends upon the data and the application. So these slides I am showing because for our Devo graph work, which one we should choose. For that, I have shown this one. So as I have shown already, there are two kind of temporal graph: discrete time and continuous time. So for them to process using neural network, what kind of we need some temporal GNN. So here I have shown both discrete time temporal graph and continuous time temporal graph. So this is a time we have taken a snapshot at different timestamp, and here we can use RNN to understand the time portion, and this and we can use CNN to understand its structure, to exploit its structure. So here in GNN, as I already shown, we use message passing. So here what we do, we take messages from its neighbor and then we aggregate and we update, and that will happen in a single snapshot. And then what we will do, we'll take a RNN and whatever the parameters or like embedding will get from this snapshot, we'll pass it through RNN. And for RNN, the hidden information will be these GCN parameters. And for event stream graph representation, where whenever there is a new event that is happening, whenever there is a new edge, we are using continuous time dynamic graph. How we will process? Similarly, we will use temporal sample. We will sample out of these, and then we use temporal aggregation, and then we will do temporal propagation. And based on the information, updated information, we will do under the. Then we'll have some structure, and we'll try to understand what kind of task based on the task. So, like it can be both, like edge level or node level task or graph level task, whatever that is needed. So here we can process temporal evolution and Here temporal GNN are extension of GNNs, and they aim to learn both graph structure as well as temporal dynamics. So for temporal GNN, as I have already shown this, we have for DTDG and CTDG, we have different kind of temporal graph neural network for both kind of graphs, and it aggregates information not only from neighbors but it across time, and the goal is to understand or predict the future state of the graph or classify the temporal pattern. So now I'll show an example of DTDG, temporal graph neural network of DTDG. One is evolve GCN. So how it works? So this is at different timestamp. So we have a GCN, as I already told. So we have different layers based on multi-hop information we take, and then we are running RNN here. So RNN is capturing. RNN can be GRU or LSTM. RNN is capturing its temporal features, and GCN is capturing its spatial feature or the structural feature, structure of the graphs, and it allows the GNN to adapt to the changing graph structure. This is evolve GCN. So how it works? At each timestamp, we use RNN to update the GCN's weight based on the previous timestamp. And here, what we are so these are the like GRU, that is taking the GCN's previous, what we call it, the previous information, and the graph convolution, it is taking the graph structure, and this process is repeated for each timestamp, and finally we get a node embedding at time t. So now I am showing example of a CTDG, that is DGN. We can show it as a encoder decoder kind of network. So there is a CTDG graph, and we use DGN, and suppose we need based on the previous information, we want to see if there will be any edge at T8. At T8, if there will be any edge between 2 and 4. So we run TGN, and we get the embedding, updated embedding at T8 of this edge, of this MLP or some other, and then we try to predict the probability given at T8 time. What is the probability there will be an edge between 2 and 4? So it handles event in order of occurrence, events in continuous time using message passing and memory model. So here is the broader view of this. What are the key area or key innovation here? Here they use the time encoding. So based on the time, suppose there is a like at t equal 5, there are some there, like I want to see at t equal to 5 and t equal to 6 if there is any age that exists between 1 and 2 and t equal to 6, and if there is any age that exists between 2 and 3. So what we have before, we have raw messages on like 1, that is raw message of node 1 and node 2 at time t1 t2, and raw message 2 and 3 at t3 t4, because there are some interactions that happen before, and we do message passing, and based on the message passing, we updated the its embedding at a different timestamp, and then we aggregate those, and we at t equal to 3 we get some embedding based on the aggregation. So, and we already have a memory. We have a memory module where we store the previous information of these nodes. So, based on the memory, we have some. This is the state. State is what was its information that was stored at t equal to 1, T1, T2, and T4 at that time. We have the memory. Now we are fetching the memory, fetching from memory its states, and we have the like based on now, if there is any like event that is happening, or if there is any transaction that is happening, based on that we will update at T5 what will be its embedding at T5 for Z1 and Z2. That means for node 1 and node 2. And then we'll try to, using the decoder network, we'll try to understand what will be the age probability between 1 and 2 and 2 and 3. If there will be any age probability, or if there will be any age that will happen at this time. This is how, and we based on the, we'll calculate the loss. Based on that, we can train this network. So, so C. elegans, as we know, it's a nematode worm, a model organism in biology. Its development is complex and it's dynamic process. So, based on the previous work, the DevoGraph last time, one person he already worked on this and used different graph type to model its development. So this is the visualization of it. So here nodes can be cells, ages can be cell to cell contacts, and time is development stage. So this is a visualization how it is changing. These are the applications that has been already explored. So now what I want to do is, if we can use temporal GNN to understand the development of these C. elegans. So we can represent C. elegans development as temporal graphs. It can be DTDG. At different timestamp, we'll take a snapshot of its development. And then we can use temporal GNN on top of it. So DevoGraph already have tools and data sets. Now my goal will be to predict it can be prediction of cell fates, that is how cell is changing with time, cell features. It can be like key interaction, cell interaction during its development we can identify, or we can understand the dynamics of the development process. This we can do. This can be the goal for the like next. So in this presentation, I have covered temporal graphs, that is good for modeling dynamic system and understanding time evolving relationship, where temporal graph neural network are important tools for learning these temporal graphs, and it extended already the capability of static GNN. That was not there. And applications, there are a lot of applications of it: social network, finance, biology, traffic, and it is working really well. So in production, like CTDZ, they have used in Amazon or Uber Eats or Pinterest. They already have used this temporal graph, and recently for recommendation, LinkedIn is also using temporal graphs. So for this work, the future direction will be we can use temporal graph neural network to understand the development process and the linear structure of CLA using temporal graphs. Thank you. And if you have any questions, please feel free to ask.
Thank you, Jayadratha.
Thanks.
That was good. Good. So yeah, we have any questions? So I have I guess basic question about the different types of networks. So you proposed you have discrete time networks and continuous time networks. The discrete time networks are these kind of snapshots of time, and then the discrete network, the continuous networks are these timestamps. So you're reporting a continuous process and you get a timestamp.
Yeah.
So I guess you asked which one is better for C elements, and I would argue that maybe both of them.
Yeah, we can explore. I think we can explore both of them. But initially, I believe we can work with DTDG, discrete time program, because we already have information on different timestamps. There are different timestamps already there in the data sets. So we can work on that and try to predict. And maybe my answer will be for different tasks, we'll use different network. So as I was telling here, so for predicting the cell fates, we can use DTDG. For understanding the dynamics of development, we can use CTDG for the dynamics of development perspective.
So I mean, the thing is that obviously development is a continuous process, but our data that we usually use is discrete in the sense that it's like these snapshots. So a lot of times we've tried to interpolate between them, say this is the lineage tree at this time, this is the lineage tree at that time. But in reality, it's a continuous process. So you could have like a cell division event that you can capture midway through, and it's still kind of a continuous process. We always capture the microscopy, at least like when we analyze microscopy as discrete snapshots. You get maybe a video of the process, but we usually, kind of extract discrete features. So feature selection stage is discrete. From there on, But yeah, I like the idea of using timestamps to say there are these interactions that initiated at a certain time. So, if you were thinking about cell division, you'd have like this cell that would kind of start to, different. Everything would pull apart, and then you'd have like some point at which it's like two separate cells, and then it migrates. Or you can capture migration in the same way and have timestamps to represent those. I think you'd have to do some work to figure out how to represent them, but I think that's an interesting kind of way to do that in continuous graphs. And it wouldn't even have to be a truly continuous representation, just having something to represent.
Bradley, I put a similar question in the chat.
Okay. So yeah, we had a question from Dick. Would growth of an embryo's differentiation tree be an example? So that would mean that, how would, if you had a growing tree, basically you had more cells, they were differentiating, and then you had these sort of features like what we call differentiation waves, which are these signals that propagate from cell to cell, and it affects the cellular state. And so you'd have this sort of, it's not just important to get, say, a single cell state. It's important to get the interaction between the cells. And go ahead.
Yeah, one thing I noticed is that there seems to be a difference between a graph that grows spontaneously, so to speak, and one that grows by environmental input, by something coming from the outside, which is added to the graph. A differentiation tree presumably is mostly environmentally independent as it grows. But the graphs that Jayadratha showed would seem to grow depending or change their shape depending on external inputs to the graph. That's why I asked the question. Jayadratha, any comment on that external input versus intrinsic growth of a graph?
I believe, as I have shown here once, it's like, so if there is any change of the age update, if there is, sometimes we don't need to have new ages or something new interaction that is coming up, but what if the existing for the existing network, the edges or the node, its features that has been updated that you are mentioning. So it's a intrinsic feature that is just updating. So it can capture that also. We just need to know at which timestamp or where the updation that has been happened. If we can find that, we can capture in our network through. Is it the answer you expected or something like?
Yeah, I mean, and there's like a difference in different types of processes. So like in the graphs that you showed, you know, you're going to have growing graphs where you have nodes that are added on, like if it's cell division, and you're not representing the whole process of cell division, you're just representing a node that kind of comes into existence. And then an edge that sort of describes that new relationship. And that's kind of, you know, it. I mean, you could make it different where you had like, you know, a cell that kind of just pulls apart and then has different relationships to each node in terms of the length of the edge or the strength of the edge. And so you have like these different nodal relationships and you have these different edge relationships. And kind of one of the things that's interesting about our data set is it's rooted in space. So you have like these spatial locations of each node, and sometimes those are very important. So sometimes we can have like, you know, depending on what we're interested in, we have different spatial relationships. So say we have like two cells that are dividing and those cells are pulling apart or they're migrating, and they're migrating with respect to the rest of the network. You could plot those out like just kind of as nodes and edges, but it wouldn't really capture that process. It would be like kind of cells are migrating, but I can't distinguish between like that process and any other process. Like, you know.
Okay.
Yeah. So I don't know how to say that other than to say there's kind of a spatial aspect to this as well for both the nodes and the edges. And it's kind of like the spatial deformation, the spatial changes are also important.
Okay, understood your point. So in that case, there are already geometric graph neural network that is available. So what it does is, suppose here, through GNN also we can capture that. We can just take the as a feature. We'll take the spatial the coordinates, 3D coordinates, the spatial coordinates, and we can use that. For geometric graph neural network, what we can use, just other than the structure and the node features in geometric graph neural network, we can use the what do you call it? It's 3D coordinate. It will take that as an input, as well as it takes another kind of input. like what is its body order? I mean, the angle between two or three nodes. So that also we can capture. So if that is also important, based on the importance, we'll see and we can add those as the features. Like, and in that case, it's a different process, slightly different process. It's there are few kind of methods that are already available that exploit what do you call it, the like equivariance or invariance of the graph structure or the geometric structure. Yeah, so those are like equivariant graph neural network or invariant graph neural network. So based on the application, we can design those also.
Bradley, got a question. Given that C. elegans is mosaic and seems to do, for a given genotype, seems to do the same thing each time, could we represent the 3 dimensions simply by making the tree 3-dimensional and let it grow?
Maybe, yeah. I mean, yeah, it would be the same, basically the same process over and over. So, yeah, you would do this, yeah.
Yeah, so I think if we actually drew the differentiation tree in 3 dimensions, it might meet your requirements to start representing a 3D graph for the growth of the nematode.
Right.
It's more complicated for regulating embryos because you get whole patches of cells which are supposedly similar. Okay, but the C. elegans data could perhaps be represented that way.
Yeah, we could do, that would be a possibility. So you'd have, instead of having the slices, like you have with your temporal graph example,
Yeah,
you would have like a 3rd dimension where it would just kind of go down, and like you'd have like the spatial aspect on the 2D plane, and then the temporal dimension on the 3rd dimension going down. So it's like time and space.
I'm thinking more like a movie where you watch the tree grow, and because it has a specific structure in nematodes, you might be able to capture it.
Oh, okay.
So it would be a movie, a 3D movie.
Right.
Okay. So time is your 4th dimension there, and you'd watch it grow, just like the one on the screen now. You can watch it grow over time. But you can imagine the nodes are represented by the actual locations of the cells and are therefore 3-dimensional.
Oh, I see. Okay.
Okay, so you'd have 3D snapshots over time, which give you a movie, basically, of a moving thing that's growing.
Yeah. Okay.
And it might fit in this framework too. So maybe there's some things we could understand about it.
Yeah. So it would be like treating those slices as a flip book, like you do with an animation.
No. I'm saying skip that stage of the slices and go straight for 3D. So when a cell divides into 2 cells in a nematode, it has taken 1 node and made 2 nodes out of it, or added 2 nodes to the lineage tree. So it's the lineage tree plotted in 3 dimensions, in other words.
Oh, okay.
Okay. And now that would seem to be maybe difficult, but straightforward.
All right.
Okay, because you'd have to get the data on the 3D locations of each cell. And and what kind it is.
Right.
But then you could make a movie and watch the whole nematode grow as a differentiation tree or a lineage tree. The ordinary lineage trees are all flattened in 2 dimensions. What I'm saying, instead of those flat ones, make them 3-dimensional. Okay, and make a movie out of it. And if that hasn't already been done, it could be quite interesting.
Yeah, that sounds good. All right, well, yeah, Jayadratha.
So,
Oh, go ahead.
yeah, is it some kind of 3D point cloud structure? I mean, in 3D, and then we'll see how it grows. My point is, how important is the spatial, like the location,
In
the
an
exact
embryo,
location?
an embryo is very important.
Yeah, we have, fairly specific data. And, we have the relational data is actually more important. I mean, because in C. elegans, the lineage tree is such that you have the spatial organization, anterior, posterior, tail, and then left to right. So there's specific relational things about different cells, and they're always basically the same in different organisms. So we have a good nomenclature. So that's important to capture. We have data on kind of specific positions. That's kind of that can be, they have a lot of technical noise. But basically, space is really important, at least in C. elegans, to get that, that sort of basic layout. So if something happens anteriorly, posteriorly, you can capture that propagates through the tree, however you represent it. So,
Also,
go ahead.
you could also represent cell motility by the nodes in the 3-dimensional lineage tree moving around a bit.
Yeah.
That might create new neighbors, which would be represented by maybe edges of a different color, because they're not descendant edges.
In that case, yeah, as I was telling, geometric graphs can be, like, we can use that also as well. And there are a lot of research that has been done already. And like, I know a little bit about that on geometric graphs, and on top of it, the geometric neural network. And there we use like, not only the spatial structure, but its actual position of the spatial position. So for drug discovery or material discovery or material property prediction or molecular property prediction, where the actual cell position or actual atom position is very important in that case. And what is the like angle between 2 atoms, that is also very important. So there people are using, but I'm not sure how, if like, since molecule is a static graph, static structure, there we can use that. But if there is any temporal like temporal part is also available or not with 3D position, that I'm not sure, that I have to check. But if we want only, if we are thinking about, or if we want only to exploit this the location or the exact position in 3D, that for that there are a lot of good models that are already up.
Yeah. So I think in terms of like a project, we've thrown out a lot of ideas here, but just to kind of focus on like one main thing you want to get done, and then you might try other things. I don't know how you want to structure the project. I mean, when you write your proposal, the timing is important. You have to get kind of have like a deliverable at the end. So, you might pick something that's kind of implementable and then experiment with some other things. So, like if you said you wanted to build a continuous time network with, some features of, I don't know how you want to structure it, but basically you could at least have one deliverable and then, go a certain distance on some of the other things and see how they work out. Now it's true for everyone's project. You don't want to just kind of propose that I do, like 50 different things and then get nothing which is deliverable. Like I know technically you could do that, but the way I usually like to have people do it is that you have one thing that you can, put together that works when you finish the project, and then have things that we can follow up on later. We have our EvoGraph repository. We've been kind of moving in varying directions. So like, each project kind of pivots away from the basic, graph neural network idea in different ways. So yeah, I would like kind of fix on maybe like thinking about maybe a continuous graph representation and maybe thinking about a 3D component and then thinking about kind of how you would put that together. First you have to implement it, of course, and make sure that you have a good framework. And then we can like get into some of the more like, some of the more biological details of it, I guess. So that's what I would suggest for the project itself. And then, we would, keep kind of thinking about it and getting specific on how to implement certain things. But yeah, like, because this is like, one of these things where it's been done in other things that are not biological, like you mentioned, social media or finance or something like that. And those are different types of systems. They have different properties. So,
yeah. We don't worry about the spatial location very much.
Sometimes, but yeah, the point is yeah.
But a customer is a customer no matter where they are.
Yeah.
Okay. But in the case of C. elegans embryos, the cells have specific locations in space in addition to time.
Yeah.
Okay. And they might also move around a bit. Now, I have a suggestion for that. When you do computer graphics, it's always on a flat screen. Okay, because we don't have 3D displays yet. Okay. Someday maybe we'll have the Star Trek hologram, but not yet. Okay, so if you had a three-dimensional differentiation tree at a given stage, on the screen it would look like it was, you'd have to flatten it to see it. But there's an interesting optical illusion that works very nicely. If the representation on the screen is a representation of something that's rotating, then our eyes see it as three-dimensional. So what I'm suggesting is a very specific project. Make a 3D growing differentiation or lineage tree of C. elegans, and as it grows, rotate it also so that it looks three-dimensional. And that would seem to me to be a project that could work given the data sets you've got. Okay. So it's a combination of making the 3D tree, but also keep rotating it so it looks 3D.
And other thing, the first goal I mentioned was predicting in future timestamp of cell fates. That is, we already have the data, the data set we already have, and those are like we can take snapshot based data we already have. So at different time, t equal to 1, t equal to 3, till t equal to 180. From minus t equal 19 or 20 to till t equal to 180, you have snapshots. So what if we can take, I mean, the simplest thing will be if we can predict, so we will train the data we have till t equal 150, and then we'll try to predict what are the future cell, features. Cell
But we already,
features.
but we already have that data. What I'm saying is, for a given, as a lineage tree develops, you know when a new node appears, so you have the timing and you have the three-dimensional position. And so you can make this graph grow over time and watch it. And if cells are interacting, that can be represented by their juxtaposition, by their being near neighbors of each other, at least physically interacting. Yeah. Because if you have two cells near each other, they can't get any closer than the size of the cells in the sense.
Okay, so it's more like the visualization kind of thing, not any prediction task involved, right?
Right. But I'm saying you can start with that, and then if you did it for different mutants, you might be able to get some different kinds of understanding. Nematodes, there are many mutations of nematodes that are known already. I don't know how many have their own lineage trees, but Bradley probably knows that.
The lineage tree for different mutants are usually just removing cells or modifying function. I mean, they don't really have their own. There aren't mutants that have enough changes in the lineage tree to make its own lineage tree. It's usually just like cells will not divide or they'll not be viable. So there's some mutants where you have a slightly fewer number of cells than the adult. Yeah.
Okay.
So you could just ablate cells, and that's interesting. But it's like you could just put that into the basic structure.
Yeah. You could compare. You could have two, let's say, a wild type and a mutant, and have them both rotating and watch the consequences to the differentiation trees as they develop. Yeah. Okay, and you can have two rotating simultaneously on the screen. I think it's time to stop viewing embryogenesis as a flatland thing.
Oh, yeah.
Okay, so you guys have all the skills and all the data, so I think it's just a matter of doing it.
Yeah.
Okay. One more thing I wanted to ask. Which data I can use in the DevoGraph? If you might take.
We have data sets. I have to pull up the data sets, and I also still need to get data sets, or I still need to provide links to the past GSOC projects, which I'll do, and I've not done that. But yeah, we have some data sets. I'll make them available. I'll put everything in the Slack channel if everyone has access. I don't know if Jayadratha has access to the Slack channel.
No, actually, I still don't have that access yet.
Well, I think I have your email, so I can go and send you the links, and then also make them available in the Slack channel.
Okay, Bradley, if there is data on syncytial features, maybe throw that in, because.
Yeah.
Okay.
Yeah, I don't know. We talked about that last time, and I haven't followed up on it, but I don't think there are any good data sets for syncytial cells and C elegans.
Okay.
Yeah, I wonder if it could be extracted from the lineage tree, though, because we do have the lineage tree where we have the interactive lineage tree.
Oh yeah, and you know which cells are supposed to be syncytial.
Well, we have some. Yeah, we have from the papers. We know kind of certain parts of the lineage tree that are supposed to be centricial.
Yeah.
So you could
Yeah.
do like you could build a database from that, I guess. Like you could just say this cell and this cell go together, this cell and this cell go together. So just identify the cells that are centricial, and then we have the spatial information, so we could just say like these are the positions for.
Yes.
Yeah.
Okay. Jayadratha, did you get that, or you need an explanation?
Yeah, a little more explanation will be better.
Well, for the centricial stuff, that's something I could work on. But like, and we can talk about that as we go.
Okay. Yeah. The problem is that the C. elegans lineage tree is usually represented as a large but simple binary tree, where you get branches at each from each cell. But what actually happens, and some people say there are 44 cases of this, some of the cells, when they physically meet, fuse together and become large cells with multiple nuclei. And that's what syncytial means in this case. But nobody's ever represented that in a graph, as far as we can tell.
Okay, understood now.
Is that a reasonable explanation?
Now I get it.
Okay. I mean, there's an example in your own body. For example, every muscle cell in your body is actually syncytial, in that it's created by the fusion of, what do you call them? I forgot what you name them, but the fusion of cells that meet and become one large cell, and with multiple nuclei in it. So it's a well-known phenomenon, but usually ignored. Complicates things because trees become full graphs as a result.
Okay.
So yeah, that's very good. Thank you for that presentation, Jayadratha. It's very good.
Yeah. Thank you.
So I don't know. Yeah, I don't know if anyone else had anything they wanted to share. You're welcome again to present on something if you have it. I know that Lalith had some ideas. I don't know what the status of that is.
Yes, sir. Last time I was discussing about like extracting the points from the movie. So maybe I'll just present my idea.
Okay. Yeah.
So I'll just give a brief understanding of what I have understood and all. So is my slide visible?
Yes.
Yeah. So in the development of C. elegans, the embryogenesis. So it starts from like a single nuclei and slowly it develops. So then we have this, what I initially wanted is in order to get a 3D segmentation model, which can actually identify the nucleus, and the exact 3D point location of it. So that from this, we can build a 3D graph. So as I have read the paper that you have written, so I understood certain key components in that. See, I have seen, there are like two graphs that can be built on top of, while segregating the hypergraph. So two hypergraphs can be built. So first hypergraph could be a spatial, so which can be arranged by physical axis. That is, it can distinguish, suppose here is the cell. From here, it's trying to grow and into number of cells, and then it becomes a full adult. Before that, all this cell has its own, and each cell, while the division happens, there is like a full lineage tree data. So we have the 3D, locations too. So what we can do is, first we can build a spatial hypergraph. So what it does is, so it extracts the cell clusters, which actually form, suppose like tissues, or we can see them, I mean, like how it forms through a cluster of cells forming a tissues. And whereas we have a linear hypergraph where it understands the temporal like structure between like hypernodes and its functional distinguished characters. And thus we can build the complete tree with it. And then we can merge these two hypergraph in order to do a full end-to-end simulation of how the embryogenesis happens. And
Lalith, can I interrupt a second?
Yeah.
It seems to me what we were talking about earlier is a third type of graph. Could you put that slide back on?
Yeah, sure.
Okay, you got a lineage hypergraph and what you call a spatial hypergraph, but you notice that it's arranged by the physical axes anterior to posterior. Okay?
Yeah.
This third type, which is a spatial graph, hypergraph, which is arranged according to the actual locations of the cells being the nodes.
Yeah.
In the locations in 3 dimensions.
Yes.
And I'm wondering why you don't consider that instead of the anterior posterior.
I mean, like, I'll get to that actually. So when we are developing this linear hypergraph or temporal construction of the tree, we can actually give this, features such as the 3-dimensional location. And in parallel, we have to feed the, the lineage tree information. How it has been split.
Yeah.
So when we combine these two, my idea is to combine them both into like the same itself. But when I have distinguished between what gets into like hyper-edge and hypernodes, so I have seen that hypernodes models the cell categories. Suppose we categorize into like saying, okay, this is the mother cell, and from this, these type of mutations happen. So if I keep the data of the mutations and the 3D locations, and I'm not really sure whether I have to keep a 3D location or keep the mutant information. Which it has mutated. And the hyper edges, we can, model it as like an anastomosis. So where we can see that, because hyper edge does not give, it do not have to be, certain these 3 nodes or these 4 nodes. So it can have any, a bunch of, hyper nodes. It forms an hyper edge. During that, it can bring some cross connections. So I am still figuring out how do I have to, pipeline this both into a same. So, but what my idea is to first is to understand, build a lineage hypergraph. So where it models the information and brings a lineage hypergraph tree, and spatial hypergraph tree too. So where, we can merge these two into a single, hypergraph neural network, which can.
Yes, but I'm just saying that the spatial hypergraph would be more accurate in a sense if you used not the anterior posterior location, but the actual 3D locations of the cells and use that as nodes.
Yes, definitely I can do that. Not just the anterior posterior, we have like 3-dimensional coordinates, so we can plug in that too. I just mentioned it as an example.
Yeah. Now the other thing, I like the idea of anastomoses. The anastomoses can be of 2 types. One is cells that are merely neighbors being represented by an edge, maybe an edge of a different color.
Yes.
Or cells that fuse, which would be the syncytial cells.
Yes, because there are suppose we are supposing this as a network, then it can be a muscle based network, or the neurons that actually activates the muscular movement, and that actually gives the information about the food or the predator. So these two, which might not be physically connected, these can be cross-connected using the hypergraph. That was the intuition from that paper I got it. So I thought this can be done by tweaking some parameters in designing the hyper edge.
Yeah,
so,
in displaying it, I'd suggest just using different colors for the edges as to whether they represent the original descendant edges in the lineage tree, or if they represent juxtapositions of cells or fusions of cells.
Okay.
Okay. And
Yes.
can you think of anything else, Bradley?
Oh, yeah, in this slide you say for anastomoses, non-tree connections. So the anastomoses are really kind of like, say you had two subgraphs where the cells were located in space, and the idea is that either the cells migrate so that they form these kind of clusters, and then the anastomoses are kind of
Relation.
the weak connectors.
Weak ties, yes, sir.
Yeah, so they're kind of like, I don't know, they're not really non-tree, they're kind of like, I don't know how to describe them, maybe weak connectors or, cross subgraph, linking subgraph, something like
Yes.
that. Yeah.
Yeah, no, I'm not sure that a nerve cell attached to a muscle cell and activating the muscle cell is weak.
Well, that's the terminology they use.
Yeah, yeah.
Yeah.
But it's but they're still separate cells.
Yeah.
Whereas if they are syncytial, then that would require different representation.
Yeah.
But I think you can get that. Perhaps you can get these things merely by changing the color of the edge.
Sorry, I didn't get it. I'm sorry.
Okay.
Okay. Yeah.
So, I mean, like, the data that is available in the DevoWorm, and I think DevoGraph has the data, both in the Mehul's repository and the DevoGraph repository. I can take the temporal locations and model a hypergraph, which can, first I will try to model this linear hypergraph and spatial hypergraph and see for a certain short movie where it can, merge, it's developing from small cells to multiple cells. So how the chains that look like, and probably I can give some more insights, or maybe I can bring up a new, a detailed direction of like what could be done from this. I'm also interested in like doing, automate this process, because this process takes a whole lot of time for like, to manually annotate from like each and every cell. So if we can, make this process like quite automate, that's already there in DevoLearn, I think.
Yeah.
It has been already done by, but we can bring more advanced methods where which can do it like more with more precision, like 98 or 90 plus 95% plus accuracy, so that this automation could be more stronger enough to deploy it in the real world.
Yeah, Lalith, if you just take these pictures in your second row, the bottom row, suppose you draw lines between cells that descended from another cell.
Yes.
Now, I'm not sure exactly how you'd represent that, but because in a way, it's a line in time between, let's say, the leftmost picture and the next one. Okay. In other words, some of the four cells on the left-hand side gave rise to two new cells, and the line goes from the line between them, or the edges between them. There would be two edges. Would go from the left image to the right image.
Yes. Yes.
Okay. So it's not clear how you're going to make that clear as a display. Okay.
This is just a representation of the image. Like when we actually play the actual video, then it starts from like two cells and then it multiplicates into that. So.
Yeah, but you're not. What I'm saying is the relationships between the cells are not shown here.
Yes, I mean, in this diagram, we do not have any relationships mentioned. Yes,
All we're seeing is a projection of the 3D locations.
yes, it's only displaying the 3D location.
Well, even
Yeah.
then, it's two-dimensional display of the 3D locations.
Yes, I mean, I can make a 3-dimensional new, good display of 3D next time. So this was just, I brought it from that paper, which I have
Well, the
cited.
question is, how do you do a 4D representation with time?
These, in this work, what they have done is, as this is a 3D data, they considered this each node, and at each node they embedded the information of the lineage tree. So doing so, they said that it's a 4D, because in a 3D structure, that each cell type, what they have, and they have the tree information too in that node. So that's how they say that this is a 4D model. So my focus is not on 4D model as of now. So my focus is on 3D itself. But
For us mere humans, I think the fourth dimension is going to have to be time, which means that the proper representation will be some kind of movie.
I get the point. Yeah, I mean, during the fourth, when we mentioned the time is the fourth dimension, while moving, in this scenario, they haven't considered the time as a fourth dimension. These people have considered that the information that persists inside the cell nuclei is there one of the feature. So when it drops down, this is a four-dimensional, three-dimensional structure which has three-dimensional location, and at each three-dimensional location they had an extra feature called the lineage information. So thus it makes four dimension for them. So if you consider the time dimension too, it will become fifth dimension probably.
Yeah, I mean, one could imagine, for instance, that if we had enough colors, that each cell type is represented by a single color, and when the cell divides, its daughter cells are temporarily represented by a separate color, so you can see where the daughter cells are.
Yes, that can be done.
Yeah, that's an example. I mean, it's not. That's why I say it's not obvious how to represent this, but I think some sort of either using color or time as the fourth dimension might make this much easier to understand.
Yes, I get your point. I mean, the way, we model suppose we model the whole embryogenesis using some a neural network like hypergraph and all. So the way we visualize, it actually makes more sense when we visualize based on the intuitive way that we have approached with.
Yeah.
So, but I just bought this from this paper, so I cannot, So next time I can bring a nice, good presentation out of that from the data.
Okay, so maybe we're on our way to getting a proper four-dimensional lineage tree.
Yeah.
Okay.
So yes, sir. I mean, this is what I'm thinking of. Probably I'll come with a more refined presentation or a refined documentation of what could be done and how I could, keep a nice good checkpoints and detail what could be the plan of action for this couple of time. And
okay, maybe you and Jayadratha should communicate with each other about this too.
Yeah, sure.
Because he's been looking at the temporal aspect, and now the question, in a sense, is okay, how do we represent all of this so that we can make sense of it?
So I mean, my one question is, can I start with developing an automated segmentation model for the cells and then apply hypergraphs, or shall I start from the 3D point locations which we already have?
No, actually, those are four-dimensional.
Yes, sir.
Because an individual cell can also migrate within the embryo.
Yeah, I mean, four-dimensional information.
I know. It's hard to put all this together.
we have the data. I would start from the extracted data that we have, so the numeric data.
Yes.
We can think about the features selection capabilities later. I mean, like,
Okay.
we have, because DevoLearn, the history of that is like we've used a number of methods with it. So, as the methods have improved, and it takes, it's a pretty fast generation time. So we've worked with pre-trained models, we've worked with different types of convolutional neural networks. We've then moved to the segment anything model. I think that was like 2 years ago. So we have all this stuff in the DevoLearn package. And, if it's something that you can think of a good, improvement to segmenting images, then, I'd work on it, but I wouldn't start there because we have data, that numeric data we can just use to see what
Yes.
we can do with, because, we would want to use the DevoLearn stuff on microscopy data. But a lot of that, we've had this pernicious problem of trying to sort of segment membranes and only membranes, and segment, nuclei and only nuclei, getting those right every time, and it's very challenging. It's not an easy problem, even in C. elegans. So
Maybe Grok will do it for us.
yeah, I mean, I know the segment anything model keeps improving. So I don't know what the current fork is. I mean, but yeah, the idea is that we want to make it very, very, if we want to go in that direction, we want to make some improvements that don't kind of reinvent the wheel. We just want to kind of improve upon the performance. Yeah.
Sure. So, I mean, siding out to the point, so after SAM, there is a new version called SAM2, which actually is good for the, the temporal version of the images. I mean, videos.
Yeah.
So, okay, sir.
Great. Yeah.
Thank you for that.
Bradley, I got a question. Is there enough data to take a given wild type, let's say, and look at variation in it? In other words, are there enough data sets so we say, this is the location of a cell in this embryo at this time, but it's slightly different in that in another embryo at the same time?
Well, I think the data sets that people have collected, and they collect them over a number of different embryos, and they just average them together. Because a lot of that variation is stochastic. It's kind of intrinsic stochastic variation. So there's going to be some variation in the exact position, but relatively they're usually in the same position. So one, cells are always anterior to other cells, but their distance, is going to vary by a little bit, and it's right.
But in other words, can we quantitatively ascertain how good the claim is that for a given genotype, a mosaic organism ends up being the same?
You mean with their relative position?
Yeah.
I think that's...
Look, this goes back to the problem of fluctuating asymmetry in regulating embryos. Why are they reasonably precisely bilaterally symmetric on the outside? Are there feedback mechanisms that correct for errors? And you could start to ask that question in nematodes, because this assumption is they're all the same, but actually you're saying they're not.
Yeah, there actually are mutant phenotypes where you have I said that sometimes cells are ablated, but sometimes the phenotype is sort of different. It's like blobby versus there are other different attributes to so that fluctuating asymmetry, given that I guess the idea is that the environment has some effect, or that there's some effect in how the cells
but
position.
I'm asking, assuming the genotype is the same between two individuals.
Yeah.
Can we start to get at the question as to why they're so similar? In other words, are there feedback mechanisms that correct for minor errors?
Maybe. I'm not sure.
Maybe. I'm not sure either. You know.
I mean, because it's like, how would you say, well, something is really similar versus.
Yeah, let me give you an experiment. Do you have any children's cubic blocks?
Not on me, no.
Not on you. Okay. If you make a tower of them, eventually it falls over.
Yes.
That's because there's a small error in putting each block on. But if you had a feedback mechanism to make them more accurately placed, maybe you could get it higher.
Yeah.
Okay, so it's a simple analogy, but how much deviation can you stand, and do you correct for it or not? I think that question could be asked about nematodes.
Maybe. That sounds like something you'd have to simulate. Like,
yeah, maybe
try different things, like different distances and tolerances and things.
Yeah, until you get a nematode that falls apart, right?
Yeah.
Okay, but if you can get some kids' blocks, try the experiment. You'll see exactly what I mean.
Yeah.
Okay. Because it always falls down eventually. But that's because kids don't make any corrections as they put them. If you made a stack and it's still stable, you could then push it together and make it even straighter so you could get it even higher. Okay, but is there a mechanism that does that?
Right.
Okay, so I think that's a fundamental question in embryogenesis, which I've never seen addressed.
Yeah.
Okay.
Yeah, that's all pretty good. All right. Well, does anyone else have anything?
Hi, Bradley. Hello, everyone. So I wanted to tell a few updates. By the way, I missed the Joyce presentation, but we have discussed it already in much detail. But I think it was great stuff which he's presenting. So Bradley, Soham and I are exchanging a few messages on LinkedIn for some time. So I'm not sure whether Soham has already discussed it, but so I prepared a small doc for what might be possible for his GSOC idea. So I'll just share my screen.
Okay. Yeah.
Start camera. Where is Okay, how do we share the screen here?
Should be an arrow, a box with an arrow.
Yeah, I think I got it. Share the screen. Okay, it's there, right?
Yes.
Yeah, so as much I understood, Soham is interested in more of visualizing aspect of DevoGraph and Hypergraph. Yes, he did mention other points which he want to focus on, more of research, but that is what primarily he was looking at. So I just prepared a doc which I think could have helped. So I've already shared it with Soham, but I just wanted to put it in the meeting also. So I mentioned few directions and what other tools which can be utilized. So I started with why do we need it and what hypergraph is. Skipping these parts, there are some interesting tools and libraries which we have already used before and some new ones. So the Hyper-NDPs, this is the one which I used last summer extensively. So it's built on over NetworkX, and this library extends traditional graphs methods for the hypergraphs. It provides good visualization methods, including the dual hypergraph. So what that basically means is that we first convert representations to bipartite, and then dual nodes with hyperedges and hyperedges with nodes, something like that. We discuss those points in the end of the my project. And then there is one nice library which I saw, HGI. So I'll just open that screen. I think this one. Yeah. So this was also providing a few visualizations for hypergraphs. This does not focuses just on hypergraphs, but it has graphs, hypergraphs, simplicial networks, and so on. So it is much more extensive than the Hyper-NDPs. So I think this was like interesting if you want to extend it in future. Then I was looking, so like Soham said, like what are the places that we can fill, like what are the dots we want to connect. So I was thinking, like if he plans to develop like a complete library for visualizations, like common to both C elegans and hypergraph, then having more control over it like would be like really great. So I was just seeing what are the other libraries. One is d3.js, so this is used a lot by like companies, more of like, so these are to develop for these are for developing web-based interactive visualization. It is JavaScript based, so it needs a lot of manual coding, but the good point is that we can offer, like what it offers is, it offers like really customized representation. So I did open it somewhere. Yeah, so it's like really good, but not specific to hypergraphs. Then I was seeing few more. So there is something called Cytoscope. This is actually a web-based application, really good for networking. Let me just open it. So it also provides like different kinds of graphs, including hypergraphs, and it is mostly web-based. And it has like really interesting like functions like which can be used. I think this was like really good for network, and over this library can be built for the hypergraph and C elegans. And Jeffy is also like similar for that. So Jeffy doesn't support hypergraphs. I was seeing one of the feature, like when will they support hypergraph? This was like a thread 4 years back. They still don't have hypergraph feature. I was looking at the Jeffy library, but I just like put it for the sake of putting. Then like the possible outcomes, what I was thinking is, so like for at least the visualization part itself, like different libraries can be integrated. So for example, probably XGI or HyperNetX, one of them can be taken to make a more front-end specific interactive like visualizations. So for that, either D3 or Cytoscope can be used, like one of it with one of the HyperNetX or XGI. So this is for the implementations perspective. And then, this can be deployed on like either Cytoscope or the D3. Then I was like seeing like few like research on like more of visualizations of graphs and hypergraphs, starting with graph layout and design. So like we discussed, right, dual of a hypergraph and bipartite representation. So this can make hypergraph representation like little easier, like the dual or the bipartite. And there are some like algorithms, established layout algorithms which already exist. So I was looking at few. Then so there was like one article on Y Combinator. I found it by accident, like a few weeks back. This is like they were also proposing a hypergraph, how we can represent it as bipartite graph. And so what they said was like the pairwise connectivity, like when we have adjacency matrix, we make it to incidence matrix when we make a hypergraph. So incidence matrix have nodes and edges as like connections. So this is how they represent it as hypergraph. And they are just like discussing a few things. I just put it there. But then there are like few research like papers and things I was like seeing. So I just put it. And this is like first idea. Second idea is on top of this. Once we have established bipartite representation, positioning nodes effectively becomes crucial. So there are algorithms like force-directed algorithms. So the second one, it offers like a solution for simulating physical forces. What I mean by this is like physical forces, attractive forces draw connected nodes together, and repulsive forces push unconnected nodes apart. So in context of hypergraphs, like if we see this definition of force-directed algorithms, we can adapt these algorithms by assigning weights to hyperedges based on the number of nodes they are connected to or the strength of their interactions with those nodes. Second, adjusting the simulating parameters to accommodate additional complexity introduced by hyperedges. So we can read more about this algorithm like in wiki page, and there are like some good presentation about it also. So this was like presentation from NUS, And these guys have like a paper also on this hypergraph drawing by this force directed algorithm. So this was like really cool, and this can be used for like how like if the goal is to develop hypergraph visualization from the scratch. I'm not sure whether that is a like possibly a good goal, but like I was seeing more on the literature how it can be done. Then coming last, so clustering and community detection. So this is basically like we had this problem, right? So when we made like a very crowded hypergraph, it was really not possible to understand what's happening. So instead of that, identifying clusters or communities within like a big hypergraph can be useful for uncovering critical areas, for example, some functional modules or whatever in the biological or like CLE network. So this is where I thought like this can be useful. There was like a Nature paper I was seeing. Yeah, I did not really go into detail of it. So they were using this information compression problem. So basically trying to, so basically the goal is to have better clustering and community detection, and which further can be used to make more clear hypergraphs using this compression of the hypergraph. And then later all this information can be made for the temporal analysis. So this is like the complete process I thought can be done for making this library few things. Yeah, but like definitely this is like we can discuss more on it if this much details is required just for the or the implementation is required for like this for our goal of visualization of the hypergraphs.
Okay, sounds good. Dick had a question in the chat. Proposed project: does the standard deviation and location of a cell and embryogenesis of a mosaic embryo increase with graft distance from the egg in the lineage tree? Yeah, that's interesting. I think we have the data for it, I'm sure.
Great.
I mean, to get the actual variation between embryos.
As I said, if you build a stack of blocks and don't do anything but add one at a time, the deviation tends to increase and it falls over.
Yeah.
But in a real embryo, that doesn't seem to happen. But that suggests the standard deviation in position is corrected somehow. And I'm suggesting the way to detect whether there's a correction mechanism is to measure standard deviation versus the number of nodes or the graft distance from the egg.
Yeah.
Okay, so I think we can. The data probably has that buried in it, as to indicate whether or not there is a correction mechanism.
Okay. Yeah.
Doesn't tell you what the correction mechanism is if it exists. That's a later question.
Okay. So Mehul, are you going to make that document more widely available, or what are your plans for it?
So I have shared it right now with Soham. Like these were the points based on like what are the questions he's been asking to me. I can share the repo if it can help in any way. If it can help in maybe like anyone's proposal, I think then it will be good.
All right, yeah, you could put it in the Slack channel.
Okay, sure.
I just wanted to ask you, let me have some time.
What was that?
Yeah, I wanted to ask something if we have some time. Yeah, so for my proposal, so if I understand correctly, it will be like understanding 3D, how lineage is happening on like based on the time. It's more of kind of like a 3D movie on time, is it? Or it's more about the visualization, right?
Yeah, the 3D movie idea is more of a visualization.
And the data will be shared on the Slack, later. Right?
Yeah, I'll share the data sets. We have some standard data sets we use, and then we have image data sets, but that would require people to segment the data. So maybe using the numeric data set. But yeah, I'll share the data sets.
Okay. So based on that, then I'll write the proposal. Thanks.
So GSOC proposals are due on the 7th. We have, I think, another meeting before that, but it's just prepare for that deadline. We can have more discussions offline. And then if Soham wants to present next time, he can.
Hello, am I audible now?
Yeah, you're on.
Okay, cool. Okay, so I have a quick 5 minutes presentation. Can I just show it?
Yeah, go ahead. Yeah.
Mehul did cover one half of it. So what I wanted to cover is, so I've divided my proposal into two parts. The first one takes up the NDP project from last year. And so the idea is that the growing GNN that we have last year, it's if we can convert that into a hypergraph. So instead of these pairwise connections at every time step, we have these hyper edge connections. So the idea is that since these are hyper edges, they should be able to model the structure better than standard graphs. For example, if you see here in the third graph, you can even visually clearly see that which cell, like the lineage here is more clearly represented than the graphs on the left side. So this was the first part of the project. So once we have a hypergraph ready, we could use hypergraph embeddings and then an NDP to predict the growth. So another use case of this could be that if you want to study the effect of some modulations, we could maybe make small changes in the inputs and then see what the network predicts. Once we have these hypergraphs, as Mehul was talking, one major issue is that the visualization part of it, which he covered. So what I thought was Cytoscape, since it is a very widely used biological tool, so and it allows plugins. I could use existing libraries like Hyper-NDPs and XZI and tailor it for our needs, specifically for this condition. And then we could use it as a plugin for Cytoscape so that even wider, like we could cater to a wider audience in that case. And then again, I have a third part of the project which takes it up and converts the whole thing into a library. But then I don't think we will have enough time to cover though. So I have this in the time permits section. So yeah, that's it. Thanks for your time. Would appreciate your feedbacks.
Yeah, that looks great. Thanks for showing that. I think that's pretty consistent with what we were talking about before. So, and I think that sounds like it should be doable within the summer. So.
Do you think the hypergraph presentation, the hypergraph thing with an NDP could work better than growing networks, or should I look for other ways to enhance the model?
You can use NDP, you can use something else. At NDP, we were having problems with computational power, so simulating a larger graph, I think line like 48 nodes or something was hard. And there might be ways to get around that. You might use something different. See how that works? It's up to you, I guess. All right. Well, I think that's all for today. A lot of things that we've talked about today, food for thought and all that. So, we'll follow up on some of this next time. That's all for this week. See you next week.